Clostridium tetani is a rod-like gram-positive that spans from half to 2 micrometers for 2 to 18 micrometers. Is the anaerobic bacterium responsible for tetanus? The organism produces round, terminal spores that give it the appearance of a drumstick. Its colonies typically appear as an irregular and grayish hue film over the surface, rather than discrete colonies. The film spreads outward, exhibiting a distinct swarming pattern due to the organism's motility. On blood agar, clear zones around the colonies indicate beta hemolysis, where the bacterium completely breaks down red blood cells. It also produces a strong, rancid odor, typical of many anaerobic bacteria. What is the LAMP test? The LAMP test, short for loop-mediated isothermal amplification, is a molecular method used to amplify DNA. Unlike PCR, it works at a constant temperature, usually between 60 and 65 degrees Celsius. That means no need for complex thermocycling machines. Even though PCR has been the gold standard for DNA amplification for decades, it has one big limitation. It needs a thermocycler, an expensive machine that constantly changes temperatures through several steps. That means it requires stable electricity, a controlled lab, trained personnel, and time. For many clinics, especially in rural or low-resource areas, that's just not practical. Now here's where LAMP shines, as it can bring molecular diagnostics to places where PCR just can't go. It also replaces conventional lab culture methods. The LAMP test can be designed to detect the tetanus toxin gene, known as TET-X, or other DNA sequences specific to Clostridium tetani. This is crucial for early detection, especially in wounds at risk of infection, and helps doctors act fast before symptoms even start. The use of this technique can be connected to Leavell and Clark's second level of prevention. There are 3. First, primary prevention, like vaccination, to stop the disease before it starts. Second, secondary prevention, early detection and prompt treatment. That's exactly where LAMP fits in. It helps catch C. tetani infections early, before the damage is done. And third, tertiary prevention, reducing complications after the disease has developed. Overall, the LAMP test is a powerful tool in the fight against infections like tetanus. It's fast, simple, and plays a vital role in preventing disease progression, as it can be detected early, especially where time and resources are limited. Let's talk about what makes Clostridium tetani dangerous and how we detect it. Clostridium tetani is best known for causing tetanus, a life-threatening disease marked by muscle stiffness and spasms. But what gives this bacterium its deadly power? The answer lies in its virulence factors, the tools it uses to infect and harm the host. The main one is tetanospasmin, also known as the tetanus neurotoxin. This potent toxin is produced by the TET-X gene. Once inside the body, the bacterium releases tetanospasmin, which travels through nerves and blocks the release of inhibitory neurotransmitters. The result? Uncontrolled muscle contraction and spasms. Another factor is tetanolysin, a hemolysin that contributes to tissue damage. Its role is less well understood, but it may help the bacterium spread by breaking down cells around the infection site. Finally, flagella and sporulation also contribute. The bacterium is motile, helping it move through tissues, and its spores are extremely resistant, allowing it to survive in harsh environments like soil for years. Now, how do we detect it? The key is DNA amplification. Tests like the LAMP assay are designed to identify the TET-X gene, the one responsible for the production of tetanospasmin. That means even if the bacterium itself is hard to grow or isolate, we can still detect its dangerous genetic signature quickly and accurately, often in under an hour. The TET-X gene consists of double-stranded DNA and is highly conserved within Clostridium tetani, ensuring high specificity for the test. So what does this molecule do to our body? Well, as mentioned before, the TET-X gene encodes tetanospasmin, the tetanus toxin, which weighs about 150 kilodaltons. Tetanospasmin is made up of 2 parts, a light chain, fragment A, and a heavy chain. The heavy chain has 2 key parts, Fragment C, which binds to nerve cells, and Fragment B, which helps carry Fragment A into the nerve cell's interior. Once Fragment A is inside, it has a special enzyme activity that attacks proteins responsible for neurotransmitter release. Specifically, it blocks the release of GABA and glycine, two important molecules that normally help calm down nerve activity. Without this control, acetylcholine keeps being released uncontrollably, leading to muscle spasms, stiffness, and seizures, the hallmark symptoms of tetanus. The sample used to perform the LAMP test can be biological material like wound exudate, blood, or cerebrospinal fluid, depending on the case. Handling these samples requires sterile techniques to avoid contamination. They need to be stored at low temperatures, ideally refrigerated if processed soon or frozen for longer periods, and sometimes pre-treatment like DNA extraction is necessary to remove inhibitors that could affect amplification. The LAMP method relies on biochemical and chemical principles. It uses a set of 4 to 6 primers that recognize 6 distinct regions on the target DNA. The reaction uses BST DNA polymerase, which can synthesize new strands without the need for temperature cycling. Reagents include the primers, the polymerase, DNTPS, magnesium ions, and a buffer system. Once the reaction is set up, it's incubated at a single temperature for about 30 to 60 minutes. Amplification can be detected either visually, through turbidity or color change with dyes like Swybiar green, or by fluorescence if a special dye is added. The steps are setting up the reaction mix, incubating at the right temperature, and reading the result visually or with equipment. To ensure the test is reliable, we use controls. A positive control includes DNA from a known Clostridium tetani strain, showing that the reaction works. A negative control, using water or non-target DNA, ensures that there's no contamination. Performance controls help verify the sensitivity of the entire procedure. When analyzing results, samples that show a color change or increased turbidity are considered positive for C. tetani DNA. If no change occurs, the sample is negative. If the signal is weak, unclear, or if there are inconsistencies, the result may be considered doubtful, and the test should be repeated or confirmed with another method. Regarding sensitivity and specificity, the LAMP test is highly sensitive. It can detect even low amounts of bacterial DNA, making it ideal for early diagnosis. Its specificity is also high because of the use of multiple primers that target different regions of the genome, reducing false positives. It's primarily considered a diagnostic test rather than just a screening method because of its specificity. Recapitulating, there are several advantages. It's fast, usually giving results in under an hour. It doesn't require sophisticated equipment like a thermal cycler. A simple water bath or heating block is enough. It's highly sensitive and specific, and it can even be used directly on crude samples with minimal preparation. However, there are disadvantages too. Sometimes, if not handled carefully, non-specific amplification can occur, leading to false positives. Cross-reactions with closely related bacterial DNA, although rare, can happen if the primers aren't perfectly designed. Also, residue management is important because amplified DNA can easily contaminate the environment, affecting subsequent tests. Finally, some specialized laboratories sell the LAMP kits for Clostridium tetani, like Icon Chemical Company or local molecular diagnostic suppliers. And the cost per test can vary, usually ranging from $10 to $30 depending on the provider and whether the detection is visual or instrument-based.